Hello students, today we'll discuss about DNA replication or replication of DNA. How DNA replication occurs and what is the need of DNA replication. Then see, DNA is the genetic material of almost all living organisms except some of the retroviruses. And this DNA is replicated before starting of cell division, maybe mitosis or meiosis. In the interphase of the cell division, interphase is the preparatory phase where G1, S, G2, three subphases are there in the interphase, and in the synthetic phase there occurs DNA replication where the total amount of DNA present inside the nucleus of the cell, it is doubled. Then how DNA replication occurs? What are the enzymes involved in DNA replication, and what is the replication process, the mechanism of DNA replication, how it occurs? Let's discuss. We will discuss today about the prokaryotic DNA replication. Okay. So see, what are the enzymes of DNA replication first? Enzymes of DNA replication, one protein is also there. Okay. So enzymes, see here, first one is unwindase or helicase, then second is primase, third is DNA polymerase 1, DNA polymerase 2, then DNA polymerase 3, sixth one is ligase, then topoisomerase 1, and SSBP, that is single stranded binding protein. It is a protein which is needed to maintain the single stranded structure of the template DNA strands, means the parental DNA strands, they should be single stranded so that they can synthesize the daughter strands. Okay. So see, let's discuss the process, how the DNA replication starts. We know that DNA, it is the double stranded structure, and this double stranded structure, it is linear in case of eukaryotes and it is circular in case of prokaryotes. It is circular in case of prokaryotes. But this double stranded DNA, it has to be first unwinded, it has to be first opened up. Then opening of these two strands, it is very essential so that the two strands act as template to synthesize their corresponding daughter strands. And another thing, that the two strands of DNA, they are antiparallel, that is one is 5' to 3' directed and another is 3' to 5' directed. Then which one synthesizes the, which one is continuously synthesizing the daughter strand and which one is discontinuously synthesizing the daughter strand, and why? See, 3' to 5' running strand of DNA, supposed to be, this is the 3' to running 5' strand of DNA and this is 5' to 3' running strand of DNA. Then the 3' to 5' running strand of DNA will synthesize the daughter strand continuously, because always the synthesis of nucleic acid is one direction, that is 5' to 3'. Why that is also, I'll also disclose that to you. That is, see, in the nucleic acid strand, the first nucleic acid's 5' phosphate is free and the last nucleic acid in one strand is having the 3' OH free. So the direction is considered as 5' to 3'. And always, maybe DNA or RNA, during synthesis, the direction is maintained in 5' to 3' direction only. So the 3' to 5' directed template strand, means the parental strand, is synthesizing the daughter strand continuously in 5' to 3' directed. And this template strand is also known as antisense strand, and the continuously synthesizing daughter strand is known as leading strand. However, the other strand, that is 5' to 3' directed, it synthesizes the DNA daughter strand in opposite direction, downwards. Supposed to be desynthesizing upwards, then it will synthesize downwards. So out of the two strands, the strand which is 5' to 3' directed, it takes a loop here, it takes a turn here. I will discuss all these things in details during the process. So first one is DNA strands should be open up. This should be unwinded. How this unwinding is done? By one enzyme, that is helicase or unwindase enzyme. Okay. So let's discuss first the two strands of the DNA. They unwind. Before that, let's discuss one thing, that DNA synthesis occurs in certain points. These are known as origins of replication or ori sites. In that ori sites only the unwindase enzyme or helicase enzyme, it binds so that it will unwind the DNA. And the helicase enzyme, it unwinds so that a replication bubble or replication eye is formed. Supposed to be, this is the DNA and here, see here, a replication eye is formed. Replication eye means this is, means this strand, it will, means these two strands, they will unwind in this direction and these two strands, they will unwind in downward direction, and there will be two replication forks formed. considered. Okay, so let's discuss about one fork. Supposed to be this fork, it will synthesize the DNA in upward direction. The two strands, they will be synthesized in upward direction. Okay, and in the diagram I will show you, I will discuss, I will make you clear how the enzymes they control the replication process. Okay, so let's discuss. First one, the two strands are opened by the helicase or unwinders enzyme, and SSBP, that is single stranded binding proteins, they bind in the single stranded regions of the parent strands so that they cannot be winded again. It is mandatory, it is important step so that they cannot be winded again, and to maintain the DNA template strand single stranded, it is needed. Then what happens? DNA synthesis cannot be started de novo or instantly. Means DNA strand, the template strand or parental strand, it cannot synthesize its daughter strand instantly de novo. It needs one RNA primer. RNA primer is a short stretch of oligonucleotide of RNA which is first synthesized by the DNA. Supposed to be this is 3' to 5' directed strand, and this one is 5' to 3' directed strand. This is the double stranded DNA, then it has been unwinded and now it is synthesizing its corresponding daughter strands. This diagram indicates that. Okay, so this template strand, parental strand, it is 3' to 5' directed. Okay, so first it will synthesize the 5' to 3' directed primer, RNA primer. This primer is synthesized by the enzyme primase. In both of the strands, before synthesizing the daughter strands of DNA, primase enzyme, it synthesizes the RNA primer. This RNA primer, it is 5' to 3' directed. Then to the 3' OH of the last nucleotide of the primer, the DNTPs are added. What are DNTPs? The deoxynucleotide triphosphates, means the nucleotides of DNA are attached. Okay, so this strand, which is the parent strand, that is 3' to 5' directed, it needs a single RNA primer because it is synthesizing the DNA strand, daughter strand continuously, and the enzyme which is synthesizing the daughter strands, it is DNA polymerase 3, that is a dimer enzyme. This unit, it is synthesizing this daughter strand, that is the leading strand. Leading strand is continuously synthesized, and this daughter strand, it is known as lagging strand because here many primers are needed. See why this parental strand, it has taken one turn here, because both the enzymes, this is a dimer, means it is having two units, this DNA polymerase 3. This dimer enzyme will move in upward direction. This is the direction of movement and this is the direction of synthesis. So this template strand of DNA, it takes a turn here. Template strand means that is the parental strand. It is taking a turn here, so that it needs RNA primer, and to the RNA primer the DNA stretch is added, then again RNA primer, again DNA stretch, then again RNA primer. So many RNA primers are needed to synthesize the lagging strand. Lagging strand is discontinuously synthesized, and this lagging strand, it is synthesized in downward direction, but it shows that it is synthesizing in upward direction because the template strand is, template strand means the parental strand, it has taken a turn here just to manage the direction of synthesis. It is synthesizing in upward direction, it is also synthesizing in upward direction to just manage, so that the dimer enzyme will move in a single direction. Is that clear? Then see here the RNA primer and the short stretch of DNA nucleotide, RNA primer and DNA, that is DNA RNA hybrid, it is known as Okazaki fragments, basing upon the scientist Okazaki, who has invented that in the lagging strand many RNA DNA hybrids are first formed. Then the RNA primers are removed out. Removal of RNA primer, it is done by the enzyme DNA polymerase 1. DNA polymerase 1, it by the 5' to 3' exonuclease activity, it removes the RNA primers. Okay, see, supposed to be this RNA primer is removed, then there is a space. This space is again filled by DNTPs by the DNA polymerase 1 itself. Okay, and here also the DNA polymerase 1, it removes the RNA primers, and in that space it fills the DNTPs. Corresponding DNTPs are added there, which are complementary to the template strand. Okay, and ligase is the enzyme which is rejoining the old DNA strand and the newly synthesized other DNA strand, because RNA primers are removed and short stretch of DNA is again added there by adding new DNTPs, so the two strands should be ligated. It is done by the enzyme ligase. Okay, so lagging strand, it is discontinuously synthesizing, and ligase enzyme and DNA polymerase 1, these are continuously needed during the synthesis of lagging strand, but here only once. Okay, because a single RNA primer is needed here. one time RNA primer is needed, then the daughter strand is continuously synthesized, that is the leading strand, that daughter strand is known as. Okay. Then see, primase role, I have told you. Ligase role, that it is what? Joining the old strand of DNA and the newly formed daughter strand of DNA, so that they can be ligated. Ligase. DNA polymerase 1 by 5' to 3' exonuclease activity, it removes the RNA primers, fills the gap again. Then DNA polymerase 3, it is the sole enzyme for DNA synthesis, daughter strand synthesis. It is synthesizing this daughter strand in upward direction and this daughter strand in downward direction, but it shows that it is synthesizing in upward direction because the two dimers, this enzyme, it moves in upward direction. Then topoisomerase 1. What is the role? Topoisomerase 1 is an enzyme which cancels the positive supercoiling of DNA double strand. So that is supposed to be, this is the two strands of the DNA which are unwinded at a time. Then there occurs positive supercoiling ahead of the replication fork. This is the replication fork. See, this is the replication fork here, and where it opens suddenly, then in the ahead of the replication fork there occurs positive supercoiling. So this positive supercoiling, it is revealed, it is cancelled by topoisomerase 1. How? It creates a nick in one of the double strands so that they unwind and they come into the normal coiling portion, normal coiling type, so that again it seals, it cuts and seals, and it also continuously does it work. When this portion is over, it will again unwind this portion and again supercoiling will be there. So topoisomerase enzyme is again needed ahead of the replication fork. Okay, then during DNA replication, new DNTPs are added. See, these are the DNTPs I have shown here in the lagging strand synthesis. These DNTPs, they should be complementary to the template strand of the DNA. Template strand means the parental strand. Okay, so these DNTPs should be complementary, but certain times these DNTPs means nucleotides, they are mismatched. Wrong base pairs are incorporated. So there is need of a proofreading here. Means the wrong bases, this should be removed out and correct bases should be incorporated in that place. This is done by the DNA polymerase 1 again. It removes the primer and it converts the proofreading. How? By 3' to 5' exonuclease activity. By 3' to 5' exonuclease activity means it starts from 3' to 5' direction, it moves and it removes the mismatched nucleotides and again fills with the correct nucleotides. Okay, so this is done. Proofreading is done by DNA polymerase 1. DNA polymerase 1, as it moves in both the direction, 5' to 3', 3' to 5', proofreading activity. So this DNA polymerase 1 is also known as clinofragment. This DNA polymerase 1, it is also known as clinofragment. Okay, this is known as clinofragment. Then see, DNA polymerase 1, it is doing this work. Then what is the role of DNA polymerase 2? DNA polymerase 2, this enzyme is responsible for repair of DNA. If during DNA replication there occurs any accidental thing, so that the certain parts of the DNA they need repair, then repair of DNA, it is done by DNA polymerase 2. So all the enzymes, they are needed likewise. See, unwindase and helicase, it opens the DNA. Primase, it synthesizes the RNA primer so that DNTPs should be added. Then DNA polymerase 1, it has two activities, removal of the RNA primer and 3' to 5' proofreading. Then DNA polymerase 2, it is for repair. DNA polymerase 3, it is the synthesizing enzyme, means it is the sole enzyme for DNA replication, synthesizing the daughter strands. Then ligase is the enzyme which is sealing the two daughter strands, means the newly synthesized DNA and the old DNA strand after removal of RNA primer. This should be ligated by the enzyme ligase. Then topoisomerase 1, it is what? It reveals the positive supercoiling ahead the replication fork, so that it will be easier for the helicase to open the to unwind the strands ahead of the replication fork. Then SSBP, these are the single stranded binding proteins which bind in the single stranded regions of the DNA template strand, so that they will be unwinded, they will be remaining unwinded till synthesis process is over. So this is all about DNA replication, and it's a very interesting and very easy process, and these are the enzymes needed, and this is the DNA replication mechanism showing the diagram. Thank you, and see, like and comment if you are having any doubts regarding the DNA replication. Thank you.